Hello students, good morning everyone. Today we are going to understand T.H. Morgan's experiment. T.H. Morgan is regarded as father of experimental genetics. Why? Because he was the one who did an experiment on fruit fly or Drosophila, by crossing which he gave the conclusion regarding what exactly is linkage, when the linkage will occur, when the recombination, all those concepts we are going to, when the recombination will occur, all those concepts will be understanding. So what did he do? T.H. Morgan did two crosses. Let us understand his first cross to understand what exactly is linkage and recombination. He took fruit fly Drosophila. Now, while doing crossing of Drosophila, you need to know that Drosophila, wild refers to the, if you find Drosophila around you, most of the Drosophila having one particular phenotype. Phenotype is external appearance. Most of the Drosophila which is found around us will be having brown body, and its eye color will be red eye color. So it is called, such Drosophila having brown body color and red eye color are called dominant Drosophilas, or you can refer it as wild type. So brown body, then he took, in contrast to dominant character, he took recessive character to Drosophila. He took Drosophila for his experiment because it is easy to produce very many mutants. So one of the mutant variety, he took recessive Drosophila, yellow bodied and white eye. Dominant is brown body, recessive is yellow body, dominant is red eye, recessive is white eye. Now in a cross, to represent those genes, we use alphabets as Mendel used earlier. Tall is capital T, small t, all those things. Yellow body is represented as small y because it is recessive, so we represented it in a small letter. In contrast to that, dominant character is represented as y plus. White eye is a recessive character, white small w. Its dominant character is red eye, w plus. So plus, if I represent, it means it is a dominant character. Now in his laboratory, he took dominant male crossed with recessive female. It is shown here, male red-eyed brown-bodied Drosophila was crossed with female white-eyed yellow-bodied Drosophila. Males are characterized by presence of X and Y chromosome. Females are characterized by 2X chromosome because Drosophila show XX, XY type of sex determination. X chromosome. Now, important thing to understand here is body color and eye color. It may be yellow, white, red, anything. Genes for body color and eye color are located on X chromosome. So you can consider these genes as sex-linked chromosome. What is meant by sex-linked? On X chromosome, which is a sex chromosome, these two genes are present together. They are present very close to each other. Okay, so I have represented one X chromosome. This is the way to represent X chromosome, sort of a bar like structure, and this is a Y chromosome. Here also female 2X chromosome. Male red eye is W plus, I have written. Brown body Y plus. Y chromosome of male doesn't carry body color and eye color gene. That's why just we have represent male chromosome, Y chromosome without any genes. On the other hand, female is recessive. Yellow body, that is small y, white eye, small w. So small w, small y. Now he crossed between these two and he got F1. Based on the result, he is giving these crosses. Now its gametes. Male will produce two types of gamete because males are having two types of chromosome. X chromosomes, how gametes are formed? Through meiosis. X chromosome will separate, same thing, W plus, Y chromosome will also separate. Here female will form only one type of gamete. There may be many gametes in the crosses, but all gametes will be carrying same X chromosome. If I take another X chromosome also, it is same. X chromosome is carrying small w, small y. Now gametes fuses, there will be fertilization. Male gamete carrying X chromosome fertilizes the female gamete carrying X chromosome. Then two X will come, a female will be formed. Write the genes W plus, Y plus, here small w, small y, small w, small y. So what is that female? Since 2X chromosome female, W plus is there, dominant gene is there, dominant W plus 1, if it is there, it is red eyed. Y plus is there, so it is brown bodied. What happened? Initially in the cross, male was red eyed, brown bodied, wild type. Now female became wild type. There was sort of reversal of sexes. Now cross between Y chromosome and X chromosome, Y is written, no genes. X chromosome will be having small w, small y. Both are recessive, so yellow body, small y, white eye, male. Initially female was white eye recessive, now male became recessive. Next, usually what we will do in any, so this is a dihybrid cross because we are taking two characters at a time. In any dihybrid cross, we get F1, then we'll do the F1 into F1. So we will have to cross between F1 female with the F1 male. But before that, one more concept was understood, that is concept of recombination. That is, you know that gametes are formed during meiosis. During meiosis, there is a prophase 1 stage which has leptotene, zygotene. During zygotene, two homologous chromosomes come near each other. They undergo crossing over at packetine stage, exchange the genes between them. That is known as recombination. So in between red-eyed gene, brown-bodied gene, white-eyed, yellow-bodied gene, there can be recombination based on that. So after getting F2 generation, he is giving that recombinant. So there will be recombination. Here it is shown. Look at same male I have taken, small w, small y male, and I have taken female which is a wild type. Wild type means brown body, red eye. Now between X chromosome of male and X chromosome of female, there will be a crossing over at packetine stage. Why not Y? Y doesn't have any genes, so it is not involved in crossing over. So in between two X's, there is crossing over at one point. What happens is Y, small y goes here, capital Y plus comes to the X chromosome of male. So there is shuffling of genes as it is seen in recombination phase. So now once there is shuffling of genes between these two, now two X chromosome, X of male and X of female, come together and form one F1 generation. F1 generation which is formed due to recombination, and you can call it as recombinant. So I have written small w, small w. Now in place of small y, capital Y has come because of recombination, capital Y plus. Next, X chromosome of female contain W plus, in place of Y plus, small y has come. So same recombinant I have written here. So this is a recombinant. Female formed in first cross, brown body, red eye female is written over here. Then male, yellow body, white eye. Now what happens is, in the laboratory, two types of females are formed. One parental type. Parental type are the one who we have taken wild parent and recessive, dominant and recessive parent. So which parents have such characters, which F1 has such characters, are known as wild parent. look at here, W plus means they are brown bodied, red eyed. So it is a parental type F1, recombinant F1, parental F1. All males are parental type only because they have yellow body, white eye, which is a parental character, parental F1. Now mate the male first with the parental female, next mate the male with the recombinant female. How to mate it? Let me give you an idea. Male contain X chromosome. So first mate the X of parental female with X of male. What happens? Let us write the genes. Female is having W plus and Y plus, another it combines with the male, small w and small y will be formed. Next possibility is another X of female will combine with X of male. So let's represent another X of female. Small w, it contains small y, male contains small w, small y. Okay, now we got two females. Now there can be one more possibility. Y chromosome is there. So do the cross between Y chromosome of male with one X and Y chromosome of male with another X. So already I have written Y. Y doesn't have any genes also. Only thing we have to write two X chromosome. First X chromosome of female contains W plus and Y plus. Another chromosome has small w and small y. Now let us check the phenotypes which are formed. What kind of phenotypes those are? In the first case, W plus is there, Y plus is there. If W plus, dominant character, the individual formed is red eye. Y plus is there, so red eye, brown bodied. Sex is female, these two are males. Red eye, brown body is nothing but parental type. In his cross he got that parental type. Next, small w, small y, small w, small y, no dominant allele, so all recessive. It is an again parental character, white eye, yellow body. Third, male, W plus, Y plus, both dominant, parental, brown body. And first I'll write red eye with brown body, wild male. Another recessive male, let me write here, recessive male is white eye with yellow body. So in his cross he found out that about 98.3%. Now all these four gametes, these are called F2 generation. F2 generation, look, observe the F2 generation, all are similar to the parental type. Since it is parental type, I have written it is parental. We got it by crossing F1 parental female with the F1 male. How much percentage? Percentage was about 98.3%. Majority of them were parental types. What was the conclusion? Conclusion was gene for brown body and red eye, or gene for body color and eye color was located very close to each other on same chromosome. If they are located very close to each other, they are said to be tightly linked. So gene for body color and eye color was a tightly linked gene, they do not separate during meiosis. Possibility of separation of these two genes are very less. Instead, if the genes are on the same chromosome, those are linked genes. Linked genes travel from one generation to another generation without undergoing much separation. Therefore, most of them look like parent only. But in the crossing, he got recombinants also. Even though they are tightly linked, little bit recombination can also occur. How to get recombinant? Cross the male with the recombinant F1. It's written here, F1 recombinant female with F1 male. Same way, first cross X with X of male, second X of X female with the X of male. Already I have written, so you'll get 2 females, 2 males got by Y into X, Y into X. Now let's write the genotype. X of male I will write first. X of male contains small w, small y. First X of female contains small w, y plus. Next, second X into second X of female. It is female is having w plus. First I have written female now. Another small y. Male is having small w and small y. Next, y into x, y into x. Let us do. Y already written, no genes on y. On x, you will have small w, y plus, w plus, and small y. Now let us analyze its genotype or phenotype, sorry. Y plus is there in the first female. Y plus is brown body, but for eye color it is not dominant. Instead, eye color is recessive, white eye. Brown bodied, white eyed is not a parental type, it is a new combination called recombinant. That's how recombinants were got. Next, look at this female, W plus is there. So it is dominant for red eye color, but body color is recessive, yellow bodied female. Again, it is not a parental type, recombinant. Y plus is there, so it is brown body, eye color is recessive, white eyed. It is also not a parental type. W plus is red eye, yellow bodied, male is formed. All these formed are not seen in the parent. A new combination got formed. Its percentage was found to be 1.3%. It was due to recombination. So through this it was understood why the new varieties are formed. If two genes are very closely linked, then most of possibility of separation of those genes are very less. Most of the parental types are formed. But it depends. Recombinants formed depends upon how much closely linked they are. If they are loosely linked, if the genes are, suppose if these genes are very far from each other, you will get more recombinants. Since they were very close to each other, you have got very less number of recombinants. Okay, so this is about cross 1. Thank you. I hope you have understood this crossing. If you have liked it, please click the like button. Thank you.